We were stuck in deep doo-doo, scratching our heads, racking our brains, looking for a clue. We thought that vaults assembled inside the cell by the same process that viruses use to form their structures. It's called self-assembly. But when we tested the self-assembly model, we were baffled to find that vault assembly occurred by a completely new process that had never been described before. Fasten your seatbelts and get ready for some new cell biology. Hi, I'm Lenny Rome, the Vault Guy. My passion is a common particle of the human cell called a vault. In episode 12, I reviewed the problems we had with our assumption that vaults formed by self-assembly and the experiments that told us that we needed to completely rethink how vaults were made. Today, I want to cover three things. First, I want to tell you what we found when we modified vault structure by changing the amino acids at the beginning of the major vault protein. Second, I want to spend a few minutes describing some of the important details of how proteins are made in the cell's cytoplasm. And third, I want to tell you how we finally discovered the process of vault assembly. Stick around for some basic cell biology that is so new it hasn't made it into a textbook yet. Let's start by taking a very close look at vault structure. Vaults are made from 78 copies of the major vault protein, which we call MVP. One MVP has been colored red. I'll highlight it here. The vault is symmetric around the waist. If we color the top half of the vault green and zoom in on the waist of the vault, we can see that MVP chains from the top and bottom interact the most at the start of the MVP chain, the N-terminus. Here are the first 10 amino acids at the N-terminus. Jan decided to make substitutions in the actual amino acids with the amino acid histidine, a small neutral amino acid that is often used to probe a protein's structure. He started with the third amino acid, which happened to be a threonine, abbreviated T. He replaced the threonine with a histidine, abbreviated H. The vaults that formed were just fine. He then proceeded to systematically swap out the actual amino acids at the beginning of the MVP sequence with more and more histidines, with 2, 3, 4, and even 5 amino acids substituted with histidine. He was still able to express these modified MVPs and make structures that looked like vaults. However, something really weird happened when he substituted 6 amino acids with histidine. He saw no vaults. Instead, he saw these very large structures that looked like rolls of aggregated protein. Occasionally, these rolls looked like they were unraveling. Jan used our antibody against MVP to show that the strange structures were made from MVP. We examined the rolls that were unraveled, and it looked like 2 MVPs were coming together. We thought that perhaps these could be dimers. That means 2 MVPs. And the dimers were forming stacks. This got us thinking that perhaps the vaults were made as dimers and not as single MVP chains. Thinking back, it was probably this thought that led us to revisit our understanding of how proteins are made in cells, and eventually it led to us solving the vault assembly puzzle. Recall in episode 7, I described protein translation. I told you how translation of the mRNA is carried out by a ribosome in the cell cytoplasm. I showed a cartoon like this one, where one ribosome travels down the mRNA, reading the 3-letter codons and simultaneously building a protein, one amino acid at a time. This was a simplification, since in cells, the mRNA is covered with ribosomes, so one doesn't observe an mRNA with just one ribosome attached and moving along making a single protein. Instead, it's like an automobile assembly line. As soon as the car leaves one station, another car comes right behind. On the mRNA, as soon as one ribosome attaches to the beginning of the mRNA and reads the codon for the first amino acid, it then moves along to the next station. Another ribosome comes right after it. As each ribosome moves along, reading the mRNA codons and adding amino acids, another ribosome begins behind, all subsequently reading the same mRNA and attaching the next amino acid to make the growing protein chain. What you see are mRNAs in the cytoplasm coated with ribosomes. These structures are called polyribosomes or polysomes. When the protein is finished, the ribosome falls off the mRNA and the polysome assembly line continues to make more copies of the same protein. We uncovered a classic paper published in 1982 that showed a polysome isolated from a small fly called a midge. Let me explain what's going on in this interesting electron micrograph. We are looking at a polysome chain. The black blobs are ribosomes. I'll label them green so you can see them easier. The ribosomes are moving along a chain of mRNA. The mRNA is very thin and it doesn't stain well, so I'll draw in an orange line and label the beginning of the mRNA, the 5' end, and the end of the mRNA, the 3' end. As the ribosomes are moving along the mRNA, they are producing the growing protein chains that get longer and longer as the ribosomes travel from the 5' to the 3' end. I colored these growing protein chains red. As you can see, the growing protein chains get longer and longer as the ribosomes reach the end of the mRNA. This turns out to be a super way to think about how proteins are made. Jan decided to isolate polysomes from cells that were making normal vaults. This is what they looked like. The round blobs are ribosomes. When he isolated polysomes from those cells making the strange MVP structures, he found the structures were attached to one end. Here are several examples. From these micrographs, we developed a hypothesis for vault assembly. We gathered lots of data and developed a model. Jan, Val, and I wrote up a manuscript describing our hypothesis for vault assembly. I hired an animator to make an animation of the proposed process, which I thought would help us explain our model. The animation wasn't fancy, but it was effective at conveying the data. The MVP mRNA is depicted by this thin orange line. The ribosomes that attach to the mRNA have two subunits that are colored blue and yellow. The ribosomes attach to the mRNA and begin to translate the MVP protein chains, which are depicted as red chains emerging from the ribosome as they move along. When the MVP protein chains are long enough, they make contact with the growing MVP chains emerging from the ribosome that is coming along behind it. The chains start to form dimers while they are still attached to the polysomes. What you see are growing MVP dimers. When the first ribosome producing one of these dimers is finished translating the message, it falls off the polysome. The dimer, however, stays attached and makes contact with the dimer behind it by making multiple side-side interactions. What you see is a sheet of dimers that curve away from the polysome. By the time the 39th dimer is completed, the sheet has come around on itself, and the first dimer outcompetes the next dimer for contact with the 39th, and the vault barrel is complete, and the vault falls off the polyribosome and finishes folding. The bottom line of this model is that it predicts that the cell does not make free major vault protein at all. Instead, it makes vaults one vault at a time from the same mRNA. This model explained all of the interesting data that we had collected. And the model explained why vaults are so regular, as every vault is made from 78 copies of MVP, 39 dimers. Every vault shell is the same size and weight. This is quite an amazing process. Furthermore, the ability of the polyribosome to serve as a template for vault assembly implies a new function for this cell structure, even though it's been studied for over 60 years. We were able to get the manuscript published in the journal ACS Nano. The journal published a picture of the model on the cover. You know the drill. I'm Lenny Rome, the Vault Guy. Join me in my next video, where I'm going to start on a new direction to tell you how we went about engineering the vault. Some real nanotechnology. Please remember to like and subscribe and keep those questions coming.